Hi viewers, this is J. Swami, Assistant Professor of Zoology. Today we are going to discuss the types of immunity and innate immunity in detail. As we know, immunity is the ability of organisms to resist or eliminate harmful foreign microbes and their harmful chemicals such as toxins. Immunity is classified into two types, namely innate immunity and acquired immunity. Innate immunity is also known as non-specific immunity, whereas acquired immunity is known as specific immunity. Then you can see three lines of defense mechanisms against various infections. Out of these three, two lines of defense mechanisms are grouped under non-specific defense mechanisms, whereas the third one is the specific defense mechanisms. So that indicates that the first two defense mechanisms are related to innate immunity, whereas the third one that is related to acquired immunity. Apart from these three, if we see the first line of defense mechanism against various infections that include skin, mucous membranes, secretions of skin and mucous membranes. This gives the first line of defense. And from the second line of defense mechanism, we will see phagocytic white blood cells, antimicrobial proteins, the inflammatory response. Whereas in the third line of defense, lymphocytes, that is T lymphocytes and B lymphocytes, and antibodies are included. T lymphocytes, B lymphocytes and antibodies are included under the third line of defense mechanism, which is highly specific defense mechanism. It is somewhat related to acquired immunity. Then let us discuss the innate immunity in detail. Innate immunity is present right from the birth of the individual. This is the non-specific immunity attributed to the genetic makeup of the individual, such as the immunity of humans to many infectious diseases. These mechanisms occur in precisely the same manner, irrespective of the nature of the antigen. For example, phagocytes will engulf and destroy any foreign invader in exactly the same manner, whether it is a bacteria or virus, otherwise any parasite. Innate immunity is preformed, standardized, and without any memory. As we discussed just now, the first two lines of defense mechanism play a major role in providing the innate immunity. Let us see the components of innate immunity. We have classified them into 5 categories, namely, number 1, physical and mechanical barriers. Number 2, biochemical barriers. Number 3, cellular factors. Number 4, genetic factors. And number 5, other factors. These all are the components of innate immunity. Let us discuss one by one. Let us start with the physical and mechanical barriers. Under the physical and mechanical barriers, there are 5 things included as skin, mucous membrane, cilia, cough and sneezing, peristalsis. Let us see one by one. First, see the skin. How the skin can act like the physical and mechanical barrier to provide the innate immunity. Skin is the first barrier to prevent the entry of microbes. And skin consists of thin epidermis, and thick dermis. The epidermis consists of dead keratinized cells. This layer prevents the entry of the harmful microbes. Coming to the mucous membrane of various systems. Mucous membrane of various systems contain mucus which has high viscosity, do not allow the entry of pathogens and washes them out. Mucus trap the microbes due to its sticky nature, prevent them from the attachment to epithelium. See the cilia, respiratory tract surrounded by ciliary epithelium. These cilia move actively and continuously, thereby wash away the microbes that are entrapped in respiratory integument. Coming to see here, these are all are the cilia entrapped the foreign microbes. Cough and sneezing. These mechanical responses play an important role in expelling the microbes that are entered into digestive and the respiratory tracts. The last one is the peristalsis. The microorganisms that are entered into the intestine through the process of ingestion, they drive out by the peristaltic movements of the intestinal integument prior to their development. So these all are skin, mucous membrane, cilia, cough and sneezing, and peristalsis. They all are included under the physical and mechanical barriers. Let us see the biochemical barriers. The things which are included under the biochemical barriers are sweat, secretions of digestive tract, human milk, nasal secretions, saliva and tears, urine, interferons, complements, properdins, secretions of bacterial flora, semen, and acute phase proteins. Let us discuss one by one. Let us start with the sweat. Skin has sweat glands that produce sweat with high salt concentration, hence it shows microbicidal activity. The lysozyme enzyme of sweat digests the cell wall of bacteria. The sebaceous glands of skin produce sebum. It consists of fatty acids which alter the membrane permeability and lactic acid which provide low pH to the skin. The low pH inhibits the growth of bacteria. Coming to the secretions of digestive tract, if you observe the anatomy of the stomach, the parietal cells of stomach secrete hydrochloric acid, hence the pH is 2. Due to this acidic environment, microbes unable to live in the stomach. Then coming to the milk, mother milk contain lactoferrin and neuramic acid like antimicrobial agents in high quantities. They fight against the entry of Escherichia coli and Staphylococci. Coming to the nasal secretions, mucopolysaccharides of nasal secretions, they inactivate the viruses. Coming to the tears, tears contain an enzyme lysozyme which is antimicrobial in nature. Lacrimal glands regularly clean the eyes. Saliva, the microbes present in the mouth are washed away by the saliva into the digestive system. Saliva also contain lactoperoxidase which produce toxic forms of the oxygen and secretory immunoglobulin A. Breast milk contain immunoglobulin A, secretory immunoglobulin A. Lactoferrin is an iron binding compound and decreases the virulence of bacteria bifidus factor. This factor favors the growth of bifidobacteria which inhibits the growth of acid producing microorganisms and that helps in digestion. Coming to urine, urine contains antibacterial substance which kills microorganisms. The microorganisms that reach male and female urinary tract, they removed by acidic urine. Coming to interferons, these are soluble non-toxic glycoproteins that have antiviral activity which inhibits intracellular viral replication of cells infected with viruses. Interferons are produced mainly by virus infected cells. The main function of interferons is to interact with uninfected cells and render them resistant to infection by increasing the production of enzymes that digest viral nucleic acids. The interferons can also cause an increase in major histocompatibility class 1 and 2 expression on all cells. If they are infected, the immune system can recognize the infection and take further steps of limiting viral replication. Interferons stop the transcription of mRNA, thereby disrupt the life cycle of viruses. Interferons activate the natural killer cells, thereby protects the host body from cancers. They block the cell division in tumor cells. These are extensively used in cancer therapy. Complement or complement system. These are complex enzyme systems usually seen in normal serum that together with antigen bound antibodies exhibit a variety of biological properties, one of which is the ability to lyse the cells or microorganisms. The complement system consists of a series of serum proteins that interact in a complex reaction sequence referred to as the complement cascade. The manifestation of these interactions are inflammation, phagocytosis, and lysis of foreign cells. Coming to propertins, this group of proteins present in serum involved in resistance to infections. They cause lysis of gram-negative bacteria and inactivate certain viruses. Secretions of bacterial flora. The bacteria that live on skin produce antibacterial substances such as bacteriocin, acids. The bacterial colonies of intestine exhibit commensalism and produce colicin and acids that prevent the entry of pathogens and harmful microbes. These microbes stimulate immune system against other microbes. If at all you remove these bacteria by using antibodies, the entry of pathogens become very easy. Coming to the semen, it contains antibacterial substances like spermazine and zinc. And finally, acute phase proteins. These are one kind of plasma proteins that are synthesized very quickly during infection. They increase host resistance and promote rate of inflammation. The best example is CRP. CRP, that is C-reactive protein. C-reactive protein. The C-reactive protein that binds to C protein of pneumococcus, it binds bacteria, activates complement and facilitates phagocytosis. These all are the acute phase proteins. And see the next side heading, that is cellular factors. Under the cellular factors, we are going to discuss the process of phagocytosis, microphages and macrophages, chemotaxis, opsonization, phagosome, phagolysosome, digestion and digestion, contactual cell injury, and finally the natural killer cells. Natural defense against the invasion of blood and tissues by microorganisms and other foreign particles is mediated to a large extent by phagocytic cells which ingest and destroy them. Phagocytes were discovered by Russian biologist Metchnikoff in 1883. He was awarded Nobel Prize in the year 1908. Generally, these phagocytes seen in two forms, microphages and the macrophages. Microphages are the polymorphonuclear leukocytes like neutrophils and basophils, whereas the macrophages, they are the mononuclear phagocytes like monocytes, wandering amoeboid cells after diapetesis and fixed reticuloendothelial cells. Major function of the reticuloendothelial system is the removal of foreign particles that enter the body. So, if you observe the phagocytosis process of phagocytosis, the first step under this phagocytosis is the chemotaxis. Phagocytic cells reach the sites of inflammation in large numbers, attracted by chemotactic substances, and ingest particulate matter. And another factor which influences the phagocytosis is the opsonization. Phagocytosis may be enhanced by variety of factors that make the foreign particle an easier target. These factors are called opsonins. Opsonin means prepared food for. These opsonins have two distinct regions. One is for the attachment of phagocyte, another one is for the attachment of antigen. For example, this is the opsonin. There are two binding sites. One is for the antigen, another one is for the attachment of phagocyte. During the process of opsonization, antigens are combined with the opsonin molecule at the antigen binding site here. This complex attracts the phagocyte, thereby phagocyte get attached to the receptor on opsonin. Now, both the antigen and phagocyte brought together through the intermediary opsonin, phagocyte engulfs the antigen. The component of complement C3b, often found on the surface of many microbes. The phagocytes have the receptors for the C3b complement protein, which enhances the opsonization. The deficiency of the C3b complement protein receptor in individuals leads to recurrence of bacterial infections. This is about the process of opsonization. So, the major opsonins are, the best examples for opsonins are immunoglobulin G, IgG antibodies, certain plasma lectin, that is mannose binding lectin, C3b and its cleavage products, that is inactive C3b. These are the components in the opsonization. And coming to the process of formation of phagosome. The phagocytic membrane forms pseudopodia, surrounds the microbe and forms the phagosome. Here we can see the phagosome. These are the pseudopodia that helps to engulf the microbe and that forms a vesicle, which is nothing but the phagosome. Then coming to the formation of phagolysosome. Phagosome fuses with the lysosome to form phagolysosome. This is the phagosome and this is the lysosome. Both of them fuse together to form the phagolysosome. The bacteria are subjected to the action of lytic enzymes in the phagolysosome and are destroyed. Lysosomes secrete enzymes like lysozyme, hydrogen peroxide and myeloperoxidase. Then digestion and the egestion. The died microorganisms and the damaged cells undergo digestion due to hydrolytic enzyme action, and undigested one ejected out from the cell. And coming to the contextual cell injury, phagocytes can kill neoplastic cancer cells upon direct contact without any phagocytosis process. This is known as the contextual cell injury. Then last one is the natural killer cells. These are also called as the NK cells. Natural killer cells. These are non-phagocytic large granular lymphocytes. They exhibit natural toxicity. They kill tumor cells and virally infected cells. They are brought to proximity with their target cells by the help of their receptors to unique glycoproteins which appear on the target cell membrane following viral infection or oncogenic transformation. They have capacity to recognize viral infected cells, particularly by the presence of receptor molecules on the surface of infected cell body, which are made up of viral glycoproteins. Killing is achieved by the release of various cytotoxic molecules like cytolysine. Some of these molecules cause the formation of pores in the membrane of the target cell, leading to its lysis. Other molecules enter the target cell and cause the apoptosis. Apoptosis is a form of programmed cell death of the target cell by enhanced fragmentation of its nuclear DNA. The activity of NK cells is highly increased by soluble mediators such as interleukin 2 and interferons. These are the natural killer cells which are commonly called as the NK cells. Coming to the fourth side heading, that is genetic factors. Based on genetic factors, innate immunity is categorized into three types. Innate immunity may be considered at the level of species, at the level of race, or at the level of individual. Coming to the species immunity, all the organisms of a species exhibit similar kind of immune response against the infection of a specific microorganism. For example, all human beings are susceptible to common cold frequently caused by a virus or rhinovirus, but it is defended by dogs. Measles can attack on human beings but not on monkeys. All human beings are totally unsusceptible to plant pathogens and to many animal pathogens such as rinderpest and distemper. This immunity is something a person obtains by virtue of being a part of the human species. The mechanisms of species immunity are not clearly understood, but may be due to physiological and biochemical differences between the tissues of the different host species, which determine whether or not a pathogen can multiply in them. And coming to the racial immunity, within a species, different races may show differences in susceptibility to infection. Best example is high resistance of Algerian sheep to anthrax. Racial differences are known to be genetic in origin, and by selection and inbreeding, it is possible to develop at will races that possess high degrees of resistance or susceptibility to various pathogens. It is difficult to demonstrate marked differences in immunity in human races, as controlled breeding is not possible. It has been reported that people of African origin, Negroes in USA, are more susceptible than Caucasian to tuberculosis. But such comparisons are vitiated by external influences such as difference in socio-economic levels. An interesting instance of genetic resistance to Plasmodium falciparum malaria is seen in some parts of Africa and Mediterranean coast. A hereditary abnormality of RBC, sickle shape, prevalent in the area confers immunity to infection by the malarial parasite and may have evolved from the survival advantage conferred by it in a malarial environment. And coming to the individual immunity, this is the difference in innate immunity exhibited by different individuals in a race. The genetic basis of individual immunity is evident from studies on the incidence of infectious disease in twins. It is well documented that homozygous twins that exhibit similar degrees of resistance are susceptibility to leprotomus leprosy and tuberculosis. Leprotomus leprosy and tuberculosis. Such correlation is not seen in heterozygous twins. And coming to the last side heading, there is other factors. Under other factors, temperature, inflammation, fever, age, hormones, and nutrition are included. Let us see one by one. See the temperature. Temperature plays an important role in determining innate immunity. Homeotherms, that is, warm-blooded animals like the mammals, are easily susceptible for tuberculosis, but the paikulotherms, which are the cold-blooded animals like fishes, amphibians, and reptiles, are unsusceptible for tuberculosis. Gonococcus bacteria killed easily at 40 degrees Celsius temperature. Prior to the discovery of penicillin, the gonococcus infection was cured by the fever treatment. Then coming to the inflammation, tissue damage caused by an injury or by an invading pathogenic microorganism induces the inflammatory response. Inflammation is characterized by rubor, redness, color, heat, dolor, pain, and tumor swelling. And by all these, there is a loss of function of the inflamed area. Then there are three major events in the inflammatory response. Here you can see the rubor, redness, color, rise in temperature, tumor, swelling, dolor, pain. Finally, there is a loss of function of that particular area. There are three major events of the inflammatory response. They are vasodilation. This is responsible for tissue redness, erythema, and increase in tissue temperature. Second one is the increase in capillary permeability. Vascular permeability facilitates an influx of fluid from capillaries into the tissue. This fluid contributes edema. And from the cellular events, there is influx of phagocytes from capillaries into the tissue is facilitated by the increased capillary permeability. The accumulation of dead cells, digested material, and fluid forms the pus. Histamines and bradykinin cause vasodilation and increase permeability of capillaries. Bradykinin also stimulates pain receptors in the skin. Coming to the fever, a rise in temperature following infection is a natural defense mechanism. It not only helps to accelerate the physiological process, but may also destroy the infecting pathogens. Therapeutic induction of fever was used for the destruction of Treponema pallidum in the tissues of syphilitic patients before penicillin became available. Fever stimulates the production of interferons and aid recovery from viral infections. Fever is one of the most common manifestations of infection. Fever is caused by many bacterial products, most notably the endotoxins of gram-negative bacteria. Gram-negative endotoxins of the gram-negative bacteria, generally as the result of the release of endogenous pyrogens. endogenous pyrogens that derive from monocytes, macrophages, and include interferons and interleukins. So we will use the drugs to cure from the fever. They are the antipyretics. Pyrogens that produce the fever, antipyretic drugs like the paracetamol are used to cure the fever. Coming to the next slide here, that is the age. The very young and very old are more susceptible to infectious diseases than the rest. The fetus in uterus is normally protected from maternal infection by the placenta barrier, but some pathogens cross the barrier, causing overwhelming infection resulting in fetal death. Some, such as rubella, herpes, cytomegalovirus, and Toxoplasma gondii, lead to congenital malformations. Increased susceptibility in the young may, in some instances, be due to hormonal influence. For example, tinea capitis caused by Microsporum audoni frequently undergoes spontaneous cure with the onset of puberty. The susceptibility of vaginal epithelium in prepubertal girls to gonococcal infection is another instance of the effect of sex hormones on resistance. Some infections like poliomyelitis and chickenpox tend to be more severe in adults than in young children due to hypersensitivity that causes greater tissue damage. Conversely, hepatitis B virus infections in the newborn are usually asymptomatic because clinical disease requires adequate immune response which is lacking at that age. However, the virus multiplies unrestrained and such neonates end up as chronic viral carriers, often developing late hepatic complications. Older people are highly susceptible to infections due to the waning of their immune responses and other infirmities like enlarged prostate leading to urinary stasis. Coming to the hormonal influences, hormones. Endocrine disorders such as diabetes mellitus, hypothyroidism, and adrenal dysfunction are associated with enhanced susceptibility to infection. The high incidence of staphylococcal sepsis in diabetes may be related to the increased level of carbohydrates in tissues. Corticosteroids exert an important influence on the response to infection. They depress the host resistance by their anti-inflammatory and anti-phagocytic effects and by the suppression of antibody formation and hypersensitivity. They also have a beneficial effect in that they neutralize the harmful effect of bacterial products such as endotoxins. Elevated steroid levels during pregnancy may be related to the high susceptibility of pregnant women to many infections. Finally, the nutrition. The relationship between malnutrition and immunity is complex, but in general, both humoral and cellular immune responses process are reduced when there is a malnutrition. Cell-mediated immune responses such as the Montauk's test become negative in severe protein deficiency as in kwashiorkor. Malarial infection in the famine-stricken may not induce fever, but once their nutrition improves, clinical malaria develops. These all are about the innate immunity, components of innate immunity, and their role in causing the innate immunity. Thank you, one and all.